Hello students, this is the lesson video for lesson 2.18 and we'll be talking about proteins again, but specifically proteins that are enzymes. Here is a general overview of what we'll be going over today. We'll be going over enzymes and how they are proteins that do these four things. They speed up chemical reactions, they do not change in a chemical reaction, they are sensitive to the surrounding conditions, and they lower the activation energy. And we'll go ahead and define these things as well. So the goals for this lesson is that you'll be able to understand the structure of the amino acids and proteins. You'll be able to understand what makes up amino acids and proteins. You'll be able to understand that enzymes are proteins that speed up chemical reactions without being changed themselves. You'll understand how the environment may affect enzyme activity. And you'll understand how enzymes lower the activation energy of a chemical reaction. And remember, our overarching goal is to describe the role of carbohydrates, proteins, lipids, nucleic acids, and water in living things. And we're going to be looking at proteins, specifically proteins that are enzymes. So enzymes, to put it simply, are catalysts, which means that they are proteins that speed up chemical reactions. So typically, a reaction in your body without enzymes would take much longer than with enzymes. For example, if you wanted to replicate DNA, it would take 78 million years for you to replicate your DNA, which wouldn't allow for life to exist. However, because we have enzymes that help out with replicating DNA, we're able to replicate DNA in less than 1 day. So obviously enzymes do speed up reactions quite a bit. Do you know anyone who is lactose intolerant? That means that they cannot digest a disaccharide or sugar, lactose, which is found in a lot of dairy products. The reason why they can't digest this sugar is because their bodies lack an enzyme, which again is a protein, called lactase. And lactase is what breaks lactose apart. So you see, lactose has two sugars, two sugars, and in order to digest lactose, we would have to break this bond. This enzyme, called lactase, is what breaks the bond, and it makes it into glucose and galactose. People who are lactose intolerant do not have lactase, so they're not able to break down lactose, which can cause a lot of abdominal cramps and other symptoms from consuming too much lactose. So the enzyme lactase, again, is a catalyst, which is a compound that changes the speed of a chemical reaction without being changed by the reaction. So enzymes stay the same, but they speed up the chemical reaction. Lactase acts to catalyze the breaking of the bond that holds together the two monosaccharides. Remember, that means one sugar, galactose and glucose. Each of them only have one sugar. When they're put together, they make up lactose. Lactase is a protein, and enzymes are proteins, but not all proteins are enzymes. So enzymes are basically a special category of proteins that actually speed up chemical reactions. So as I mentioned before, enzymes are necessary for maintaining most of the characteristics of life. Enzymes are essential in order to keep organisms living, and there are many different types of enzymes that exist. So scientists have identified more than 1,500 different enzymes in the human body alone that play specific roles in the many processes necessary for life. Many more enzymes exist in other kinds of living things. So it's only 1,500 different enzymes in the human body. But if we look at another organism, they would have even more unique enzymes as well. Scientists use the suffix or the ending ase or ace to indicate that a chemical is an enzyme. So for example, we talked about lactase in the previous slide. Lactase, you see ase, is an enzyme that breaks down lactose into two simpler digestible sugars. Scientists also group enzymes based on common activities, or basically what their job or function is. So you can see some of them here. So we have amylases are enzymes that break up starches or cellulose fibers, so pretty much enzymes that break down carbohydrates. We have cellulases that remove tiny fibers from cotton cloth that make the cloth stiff and clothes dull, and prevent pilling or small fuzz balls. We have lipases, which break up oils and fats, and then we have proteases, which break up proteins. But you notice here that all of them end with ASE, indicating that they're all enzymes. So although enzymes do speed up reactions, they themselves are not permanently changed in the course of a reaction, so they stay the same. So lactase is the enzyme that interacts with the molecule lactose. The interaction breaks the bond that we're holding the two together. So for example, these two together make lactose, right? It's galactose and glucose, and they make up lactose. This enzyme right here, which is lactase, is going to break the bond that exists right here between the two sugars, so that in the end what you see is we have one galactose, and then the second sugar is glucose, and that's what the enzyme did. Even though the enzyme changes a molecule from lactose into two individual sugars, the enzyme lactase remains unchanged during the course of the reaction. So that's important for you to know. Enzymes do not change. After the bond is broken, the lactase is free to catalyze or speed up more reactions with other lactose molecules. So it's not like a disposable plate or cup where you use it once and you throw it away. But lactase you can actually use over and over and over again. And this is really an important aspect of enzyme biology. Enzymes help speed up chemical reactions, but they do not themselves change as a result of the reaction. Enzymes are essentially recycled or reused in the course of a reaction. So because of this, we don't need a new lactase enzyme every single time we need to break down a lactose molecule, but instead we only need a small concentration of enzymes because we can use them over and over again. So enzymes, another characteristic of enzymes is that they are specific. So enzymes recognize and interact with specific molecules. So we don't have one enzyme that does everything in our body, but we have many different ones that have specific functions. So again, talking about the lactase enzyme recognizes and breaks apart specifically the lactose molecule. But lactase enzyme cannot interact with other molecules even if they're other sugars. This enzyme specificity is another important aspect of enzyme biology. Enzymes recognize and bind to specific molecules. So you see here, lactase is specifically shaped so that it can fit lactose. If lactose tried to interact with other types of enzymes like amylase or catalase, you would see there isn't a match between them, so they would not be able to bind to the enzyme and be broken apart. Lactose only fits with lactase. Enzyme specificity explains why your body contains so many enzymes. You have digestive enzymes that recognize specific sugars, fats, or proteins. Some protein digesting enzymes are so specific that they only recognize certain amino acids. So again, you need many different enzymes for each individual thing that you need to break down. However, enzymes do more than just digest your food. They don't just break down things. Enzymes can also play roles in building the structural proteins that make up your body. So they can also build things put things together as well as break things down. They can also help your body quickly get rid of extra carbon. Still, other enzymes function in photosynthesis, helping plants produce sugars. The many roles that enzymes play make them an make them essential in maintaining homeostasis. Really, it's hard to describe every single thing that an enzyme does. They do almost everything in your body and really help speed up all the chemical reactions that occur in your body. And you have one that's specific to every single reaction that needs to happen. Another example of enzymes are laundry detergents. They contain many enzymes that help with the cleaning process by breaking down the proteins and carbohydrates in common stains from food, blood, and grass. These enzymes function best in normal clothes washing conditions with high temperature and high pH. So it has a very specific environment that it works best in. Now let's go over the structure of an enzyme. The structure of an enzyme really helps you understand what an enzyme actually does. Again, the structure influences its function or job. Okay, so if we were to look right here, we see that we have the enzyme, which is right here, which is the lactase, and we have lactose, which we're going to call the substrate. The substrate is basically the molecule that an enzyme is going to interact with. The thing that it's going to work on is called the substrate. And the region where the substrate right here is going to bind onto the enzyme is called the active site, which is right here. So the active site is on the enzyme and is where the substrate will bind onto. Substrate molecules bind to an enzyme's active site to produce an enzyme-substrate complex. Okay, so an enzyme-substrate complex. So once the substrate binds, like in this picture right here, we call it an enzyme-substrate complex. Once bound, the enzyme shifts its shape slightly, so it brings its peptide chains in closer contact with the substrate molecule. It's similar to how a catcher's mitt clasps a baseball. It makes it have a tighter grip on the substrate. After it binds, then the enzyme will help break the bond between the two monosaccharides, and then it'll release the product. And we would call this portion the end result, the product, and that would be the galactose and the glucose. So the specific shape and structure of an enzyme's active site determine the molecules with which it can interact. The substrate and the enzyme fit together like a lock and key. You can't just put a key into any lock. You need to put it into a specific one for it to work properly. And the same thing is true with enzymes. When bound together in this way, the enzyme catalyzes a reaction. Remember, catalyzes means speeds up a reaction, such as breaking apart a substrate molecule. The end result of an enzyme catalyzed reaction is called the product. So we see the product is right here with these two, and the enzyme is down here. These two are represent the enzyme substrate complex. In a given enzyme, the active site has a specific shape and structure. So the active site, remember, is right here, which is where the substrate will bind onto the enzyme. And only certain molecules can fit, only the molecule that the enzyme is specifically designed to work on. For example, a cellulose molecule would not fit in the lactase's enzyme's active site because the lactase's enzyme's active site is only for lactose. Enzymes also work not just to break things down, but also to bring molecules together and form a bigger product. So in addition to binding specific molecules, enzymes also hold molecules in specific orientations, allowing two molecules to form a chemical bond that would not normally form. So inside a cell, substrate molecules such as individual amino acids may move freely like particles suspended in water. Even if two molecules that could form a bond bump into each other, if the correct parts of the molecule don't come in contact with one another, then no bonding takes place. So we see here the bond is going to happen between these two sugars on these edges. But if these two sugars, galactose and glucose, are just floating around, and let's say it bumps right here, and that's where they bump into. that's not going to allow for the two sugars to form a bond. So what an enzyme does is it brings it into and orients it in a specific way so that these two portions right here can be next to each other and then form a bond. So enzymes help ensure that the correct regions of molecules interact with each other. So the correct ones, meaning like this side and this side, come close together and interact. In protein synthesis, which is when we make proteins, for example, the amino end of one amino acid forms a peptide bond with the carboxyl end. If you remember, we have different sides of an amino acid. So in an amino acid, we'll have an amino group, right? Then we'll have a carbon, we'll have our hydrogen, and then we'll have our carboxyl group, right? And then our R group. And if we wanted to get two amino acids together, we would need this group here to form a bond with this group, forming our peptide bond. But in order for this to happen, an enzyme can hold these two in the correct orientation so that the carboxyl group and the amino group are in the proper orientation that can help the peptide bond, which is right here, this is the peptide bond, that can form between the two amino acids. So how exactly do enzymes speed up the reaction? So enzymes lower a reaction's activation energy. So energy is required for all chemical reactions to take place in your body. And the amount of energy required for a reaction to take place or to start is called the activation energy. And it's like this portion right here, the uphill of the reaction graph. A system needs to overcome this energy barrier right here in order to start a reaction. So what an enzyme does is it lowers the activation energy, making it easier for the reaction to happen at a faster rate. So for example, bringing molecules together in just the right way, like orienting them so that they can form a bond, will help lower the amount of energy that would otherwise have been required to form a bond if the molecules were just floating around. And so what you see here is without an enzyme, the hill is very steep, which tells you that it takes a lot of activation energy in order to start the reaction. But with an enzyme, you see that the hill is much shorter. So you see that the activation energy has been decreased, allowing for the reaction to occur in a more timely manner. So enzyme-assisted reactions eventually will reach a maximum rate. It's not like enzymes can continue to make reactions go faster and faster and faster to an infinite level. Enzymes play an important role in speeding up many chemical reactions that are essential for life, but the rate of the reactions eventually reaches the maximum point. And that point is when all enzymes are bonded to substrate molecules. So that means every single enzyme that is present is working. At that point, we say that the enzymes are to be saturated. So because there aren't any available enzymes, no matter if you keep putting more substrate, the reaction cannot occur any faster. So here you see when the concentration of substrate is low, you see right here, adding more of the molecules can speed it up. So you see here, the rate of reaction is around here, but if we just add just a little bit more, right, we see that the rate of reaction is increasing. But eventually, when we add just a bit more, let's say we add this much more substrate, right, what you see is it's going to start leveling off, which you see in this graph right here. So even though we're adding more and more substrate, the rate of the reaction actually stays the same. And the reason why is because every enzyme is working, every enzyme is occupied by a substrate. So this right here is what we would consider the saturation point, or when the enzyme has reached its maximum rate. So although enzymes do many things, some enzymes need other molecules to work properly, and we call these coenzymes and cofactors. So I'm sure some of you guys eat vitamins as a supplement, or maybe you have different vitamins in your diet, and you might be wondering, why exactly do you need to take these vitamins? So among the many roles that vitamins play in your body, one of them is to help enzymes function. So inside the body, vitamins help produce small molecules called coenzymes. So vitamins help make coenzymes, and these coenzymes are required for enzymes to function properly. So coenzymes are not permanent parts of enzymes. They're actually just temporary. They move around and temporarily bind with an enzyme, then move on and interact with other enzymes. So certain enzymes also require other compounds such as minerals like zinc and copper. These compounds are called cofactors. Cofactors and coenzymes may help enzymes by performing functions such as moving electrons, providing energy, or binding other molecules. So normally when we had just an enzyme and a substrate, the substrate would just bind onto the active site. But in this case here, in order for the substrate to fit into the active site, the coenzyme must bind first, and then it's a perfect fit. Once that happens, then the enzyme can break apart the substrate and then produce the new products. When the new products are released, you see them coming off the enzyme, so is the coenzyme, and the coenzyme can then interact with another enzyme if it needs to. Although enzymes do many things and are very important for life, they are very sensitive, and their environment largely influences its activity. So like all proteins, enzymes are sensitive to changes in their environment, such as temperature, pH, and ionic conditions. These all need to be at an optimal level for enzymes to work properly. And each enzyme has a specific set of conditions that are ideal for its activity. Changing those conditions may affect an enzyme's activity or stop it altogether. So if it's just a little bit off, it can affect the enzyme's efficiency. For example, high temperatures may denature, or that's another word for break down, a protein like an enzyme and change its shape. If an enzyme's shape is changed, then that means its active site no longer fits its substrate. The pH of an enzyme's environment, how acidic or basic the environment is, can also affect the enzyme's activity. For example, the enzyme pepsin, which is found in the stomach where it digests proteins, requires a very acidic environment like the stomach. The enzyme salivary amylase, which digests starches in the mouth, functions at a much less acidic pH than pepsin. And so you see, different enzymes have different environments that allow it to function at its best. So we see here's an example of an enzyme and a substrate. This is the perfect temperature for the enzyme, so you see that the substrate can fit into the active site. Whereas if we increase the temperature to here, which is too hot, the protein ends up being denatured, breaks down, and changes its shape. So the substrate can no longer bind onto the active site on the enzyme, not allowing the enzyme to perform its function. So here's a summary of what we went over. We went over how enzymes are types of proteins and that they speed up reactions. They do not change in a chemical reaction, which allows us to use them over and over again. Important in recycling. They're sensitive to surrounding conditions, which can include temperature, pH, and ionic conditions. And they lower activation energy, which is that energy to start a reaction. So here's some important vocabulary. We have catalyst, which is a substance present during a chemical reaction that speeds up the reaction but is not used up or changed during the reaction. And an example of this, a catalyst, would be an enzyme. A substrate is a molecule on which enzymes act. The active site is where the substrate will bind onto the enzyme, And then we have the activation energy, the energy barrier that must be overcome for a reaction to occur. And remember, enzymes lower the activation energy, and that's how they speed up the reaction rate.